Hello and welcome to this introduction to fNIRS and NIRx. My name is Ashlyn Casey and I'm a technical support consultant in the NIRx Minneapolis office. This presentation will start with the basics of neuroimaging with fNIRS. This will cover the working principles behind this method. We'll move into considerations for fNIRS studies, advantages and applications of fNIRS, and finally product and company highlights for NIRx. This is more to give you context of the technology rather than a sales pitch. Okay, so the basics of neuroimaging with fNIRS. What is fNIRS? Well, it stands for Functional Near-Infrared Spectroscopy, and you can break that into three chunks. Functional, so we're looking at functional activation rather than structural information. Near-infrared, we're using near-infrared light to do that. And spectroscopy, which is the method of sending light into a substance and reading it out on the other side. So putting this all together, it's a form of functional optical imaging used to measure superficial cortical hemodynamics. Okay, and here are two representations of fNIRS. On the left, we've got an image of what the data looks like, specifically those changes in color that you see. Those reflect changes in the level of oxygenated and deoxygenated hemoglobin in the cortex of the human brain. The model that you see, that brain, is just that, it's a model. It could be a commonly used atlas or an individual fMRI scan, but we need to provide that information to give context to the functional changes that we see. And then on the right, we've got an image of a child wearing the cap, so we see the form factor of the technology. It's very similar to EEG, where you've just got probes that sit on the scalp level. It's safe, non-invasive, and comfortable. So what is functional imaging? Here's an analogy for you. So here we've got a storm system. This is functional activation, but without a map showing us where this activation is happening, it's not that meaningful to us. But when you add geography in this case, that being the structural information, you get more of a sense of where this functional activation is. So that's what we're looking to do with fNIRS. We see functional activation and we use our structural information to make sense of that. Okay, what is fMRI? So we asked this question to draw a contrast between fNIRS and fMRI. They do have some similarities, but are very different in terms of their form factor. So fMRI it's known as the gold standard of neuroimaging, and it stands for functional magnetic resonance imaging. So putting it all together, a working definition is that it's a form of functional electromagnetic imaging used to map anatomical structures and measure whole brain hemodynamics. So we're not just looking at functional activation, we also get structural information with fMRI, and we're looking at whole brain hemodynamics rather than just the superficial cortex. So here are two representations of fMRI. On the left, you've got an image of what the data looks like, so you get that individual structural information as well as functional activation throughout the brain. And then on the right, we've got an image of the form factor of fMRI. So this involves a subject lying down in a tube, a big mechanical tube. It's loud, it's constrictive, they're not allowed to move, so it's a very different imaging environment. But given that you get these very nice, very clear images from fMRI that include structural information, why even use fNIRS? Well, there are many advantages of fNIRS that cannot be denied. You have much more flexible imaging environment and stimuli, so it lends itself much better to a whole host of different cognitive studies. It measures more components of the hemodynamic response. So whereas fMRI, the bold response is mostly looking at the deoxygenated hemoglobin changes, fNIRS is looking at oxy and deoxyhemoglobin, and we'll get more into that later. It's more robust to motion artifact. Subjects can move with fNIRS. It can image a greater range of subject populations, so you can include awake children and infants, you can include people with developmental disabilities, people with shrapnel in their brains that might otherwise be excluded from fMRI imaging studies. It's more comfortable for subjects, and it's much lower cost. So While fMRI does have many advantages, fNIRS has certain advantages that may be more applicable to you and what you're studying. Okay, what is hemoglobin? Hemoglobin is a protein that carries oxygen throughout the body via red blood cells. So it's this protein that sits on red blood cells and carries oxygen. It has two states. It's either oxygenated or deoxygenated. So it either has the oxygen that it's bringing to the cells, or it's already given that oxygen and then it's headed back to the lungs to get more oxygen. fNIRS investigates changes in this molecule specifically. So given that it can detect changes in this molecule, that's reflective of hemodynamic activity in the cells. Okay, what is spectroscopy? Spectroscopy is the study of the interaction of light with matter, and it's used to identify or measure certain characteristics of molecular structures. So in spectroscopy, we've got a diagram of what it looks like here. We're using an incoherent light source here. It's being sent through this prism, and light is being split into different wavelengths. In this case, in this spectroscopy test, they're interested in green wavelengths. That's being sent into a tissue sample, some kind of a sample, and it's being read out on the other side. So with that difference in the light that is sent in and the light that is received, information can then be determined about the structure in between. With fNIRS, that tube there, it's not a test tube, it's the human brain. So we're sending light into the human brain to determine information about changes in hemodynamic activation. Why use near-infrared light? So near-infrared light, it's a part of the electromagnetic spectrum. It includes wavelengths from the visible red and infrared range. So it's just on the edge of visible light. And the reason we use it is that it is much less absorbed by structures in the tissue other than hemoglobin. So here we have an incoherent light source being sent into human tissue, so that's all possible visible light being sent into that tissue. And we see that the red light is what's able to pass through the human tissue. And even at relatively high intensities, high enough for detectors to detect it on the other end, light detectors to detect it. Okay, and just to give some more information on why exactly that is, so near-infrared light uses light between 690 and 860 nanometers specifically. So you've got this window here, and we see this on this curve of frequencies used in fNIRS imaging. So the reason we use them is because here we've got plotted these curves of absorption coefficients of various substances. We've got deoxyhemoglobin in blue, oxyhemoglobin in red, and then we've got bulk lipids in purple and water in green. So bulk lipids in bulk lipids in water is what mostly composes human tissue. And we see that it has a much lower absorption spectrum in this range compared to oxy and deoxyhemoglobin. So it's this phenomena that lets us use near-infrared light to investigate changes in hemodynamic activation. Okay, and one thing to note here, fNIRS systems use at least two wavelengths to differentiate oxy and deoxyhemoglobin. We're going to have two equations to solve. We have two unknowns, and therefore we need two data inputs to solve. Okay, what does fNIRS measure? We've talked about hemoglobin a little bit, but to get into a bit more detail here, continuous wave fNIRS, which is what most commercially available fNIRS systems image, uses two wavelengths of light to measure changes in hemoglobin in the blood, specifically changes in oxy and deoxyhemoglobin, as well as total hemoglobin, which is just the addition of those two. So you'll note those delta signs indicating change. We are looking at changes in oxy and deoxyhemoglobin. Continuous wave fNIRS does not take absolute measurements. So we're not looking at the absolute concentration of oxy and deoxyhemoglobin. We're looking at changes. Okay, well, why is that? Well, changes in light intensity are converted into changes in hemoglobin values using the modified Beard-Lambert law. So we take our light intensity and we plug it into this equation to then determine the changes in concentration in oxy and deoxyhemoglobin to give us meaningful information out of meaningful physiological information out of those raw changes in light intensity. So this is the modified Beard-Lambert law. I've got the everything defined here in this equation. We've got optical density, that's OD, and that is equal to the negative log of the light detected over the light emitted. And you set that equal to the absorption coefficient of your molecule of interest, so that's going to be oxy and deoxyhemoglobin. This L term stands for your source and detector distance, so that's the distance between your source and the detector on the scalp. You've got this DPF term, differential path length, so this reflects, it's a multiplication term to reflect while there is a certain linear spacing between your source and your detector, the path that your photon takes will not be direct between your source and your detector. It's going to take a bit of a circular path to get from your source to your detector, so that's what the DPF term is there to account for. And finally, this G term, which is the effective scatter. So this term indicates the exact path that a photon took to get from your source to your detector, and it's reflective of the time of flight of the photon. Well, with continuous wave fNIRS, we can't determine G. So we can determine these other terms through our setup as well as modeling, but we can't determine G exactly with continuous wave fNIRS. So we have to take the derivative. And when you take the derivative, you get rid of constants. So you get rid of this G term, but now you're looking at changes in concentration rather than absolute values. So this is very important to note. Continuous wave fNIRS measures only the concentration changes relative to a previous time point or a baseline. Okay, fNIRS terminology. So we've talked a little bit about it here, but let's just define these components. We've got a source that emits light, a detector that senses light, and you can refer to either as an optoid or a probe. So this is in contrast to EEG. A term that's often conflated with fNIRS probes is electrode. So with EEG, you've only got detectors that are detecting electrical activation. With fNIRS, you've got a source sending in light and a detector sensing light. Okay, taking a look at the fNIRS hemodynamic response. So what exactly are we looking for with our data? Well, given neural activation, we see metabolic demand, increased blood flow, and that leads to an increase in oxyhemoglobin and a washout of deoxyhemoglobin. So looking at this time course over here on the right, we've got our changes in concentration over time. So this time scale is in seconds. You'll note it's in seconds. That's in contrast to EEG, where the electrical changes that you're interested in tend to be happening on the millisecond level, whereas fNIRS, this metabolic response that we're interested in, is happening at a much longer time scale. So we've got our stimulation period. This is any kind of a cognitive or sensory stimulus. We see this increase in oxyhemoglobin in red and this consequent decreased deoxyhemoglobin in blue. You'll note that the oxyhemoglobin increases much, much more than the deoxyhemoglobin decreases. We then see the two cross over a couple seconds after the end of the stimulus period. The two cross over around the 0 point mark, and then eventually they'll return back to baseline and they'll come back together again. So this is the general curve that we're looking for to reflect hemodynamic activation in a certain area of the cortex. Okay, now let's take a look at fNIRS in action. So we've talked about some of these working principles. What does it actually look like at the end of the day? So here we've got fNIRS probes on a cap, and we've got our amplifier system that takes our changes in light intensity and it sends it to our it creates data files which we can then analyze. Okay, so this is just a short video showing us a very high density measurement, so 48 sources and 48 detectors and 16 short channels, and we'll discuss short channels more later on. But here we're making our montage in near sight, so we're placing our sources and detectors in our region of interest on the head in software. We're adding short distance detectors here. And now we have the final layout. So we've got 48 sources, 48 detectors across the head. We've made the montage in the software, then it has to be made in the actual hardware. Here we see the cap being placed on the participant's head. Okay, and this is our NIRsport 2 imaging system, and we'll discuss that more later. Okay. So you can see it's quite a few probes, but the more probes you have, if you intersperse them with each other, you can get higher resolution imaging, and we'll talk more about that later. Okay, so the cap is set up and now it's time to record the data in the data acquisition software. The first step is to optimize the signal for that specific participant. That involves setting the proper source brightness for each source. And then it's time to record the data. So this is a finger tapping task. The subject is being told when to start and stop tapping their fingers. We can see the data being projected in a couple different ways. We've got these 2D and 3D layouts as well as the time course. And in the time course, we're interested in these kind of slower, longer changes that you're seeing. This data has been filtered, so we don't see some of the higher frequency content. We're just interested in these longer, slower curves that we're seeing here. They should look somewhat like the curve that we saw on the previous slides. Okay, so that's just one possible way to investigate the human dynamic response in the brain with that finger tapping task. Using fNIRS, there's a whole host of other ways to investigate neural activation. How does light interact with tissue? Well, when passing through tissue, which is a diffuse media, light is scattered and absorbed. So scattering refers to a photon, which is a light particle, refracting, reflecting, and diffracting off of and through tissue. It all describes movement of a photon through and off of tissue. Absorption, on the other hand, is when a photon hits a molecule, is destroyed, and transfers energy. So it ceases to exist, it's no longer passing through the tissue, and its scattering and absorption is what fNIRS studies. What is an fNIRS data channel? Well, an fNIRS data channel is formed by a source and detector pair, and it's going to detect the light that is scattered from your source to your detector in between, in the tissue in between and underneath your source and your detector. You'll note that the number of usable versus total data channels can vary. It depends on your system and your setup. So for example, in the bottom right corner, we've got a top-down view of 5 sources and 4 detectors. So now you could potentially form up to 20 channels with 5 sources and 4 detectors. However, in this setup, you can see that we're only interested in channels formed by sources and detectors that are close to each other. So it's this nearest neighbor measurement. Now you could possibly arrange this so that what you're interested in is all possible combinations of source and detectors. In this case, this is a topographic measurement and you're only interested in 12 of the data channels. So it depends on your setup. This is unlike EEG, where if you have 32 electrodes, you have 32 recording channels. With fNIRS, since a source and your detector form a recording channel, the number of channels in your, that you're interested in when it comes to analysis depends on your setup. How does light travel between a source and detector? Well, light leaves the source and it travels in all directions. So that there's a representation of that here with all these different arrows kind of shooting off of the light source. Photons are then scattered and absorbed through the tissue. And there's no magnet pulling our photons from our light source to a detector. And in fact, for every 10 to the 9 photons that is sent into the adult brain, only 1 comes out at the detector on average in a 3-centimeter channel. So you have to send in millions and millions and millions of photons in order for any to be detected at your detector. So we don't know the exact path that these photons take, but what we can say is that for the photon that reaches a light detector, we can determine its average path. And that path is going to be take the shape of the photon banana. So the photon banana is the actual scientific term. It's this 3D shape that we see probing in the tissue between the source and the detector. So where does the channel measure from? As I just mentioned, it images that tissue in between a source and a detector, in between and underneath. The depth of that photon banana depends on the optode spacing. If the spacing is too long, not enough photons will reach the detector and you'll have a signal that's dominated by noise rather than your photons of interest. Spacing probes further apart leads to greater depth images, a greater depth imaged but lower signal to noise ratio. Spacing probes closer together has a shallower depth of imaging but a higher signal to noise ratio. So the exact depth of your channel spacing, it's going to depend on what you're interested in as well as your hardware. Not all fNIRS hardware is created equally, and not all can get high signal to noise levels in channels that are farther apart. So where to place a source in a detector? Well, the optode arrangement will determine your signal strength, region of interest, depth of measurement, spatial resolution, referring to both depth and surface resolution. So now would be a good time to differentiate topography and tomography. So topography, and this is the image we saw earlier, captures functional neural activation without depth discrimination. And it does so by placing your sources and detectors at equal distances apart from each other and only being interested in those channels of the nearest, formed by the nearest neighbor. This is typically done with 3 to 4 centimeter channels in adults and closer to 2.5 centimeters in infants. And that's done because of the different skull thicknesses of infants versus adults, and therefore in order to image the same depth of cortex, you need to account for that in the changing of the head size with the changing of the source and detector distance. Okay, topography is the most popular form of fNIRS imaging, so it's the most commonly done. It's simpler, it's easier to analyze, but there are certain limitations certainly in depth resolution and being able to do 3D reconstruction of images. So you're really limited to that one surface layer of probed tissue. What is tomography? Well, that's when you intentionally form channels at varied depths by spacing your source and detectors at varied distances from each other. It can measure different cortical depths, and it requires complex 3D image reconstruction. So this is a much more intensive form of imaging, certainly when it comes to analysis. So it does require quite a bit of legwork in order to produce these 3D reconstruction images. But with this 3D reconstruction, you can get a better sense of where exactly in the tissue activation occurred. So overall, this is not as commonly used in the field except for short channels. So we saw short channels earlier in that video. What are they? Well, they're used to measure the superficial scalp blood flow component in the fNIRS signal. Well, why do we care about that? Here we've got our photon banana projected onto the cortex, and we've got 3 points denoted. We've got A, B, and C. So what we're interested in probing is this B point. This is the cortical tissue. However, our light also passes through this layer of skin at the scalp level at both points A and B. So first when it enters the tissue and then when it leaves the tissue. So blood flow this blood flow measurement can contaminate your measurement from the cortex, and it can significantly impact your data. Specifically when when changes in systemic physiology occur simultaneously with changes in cortical activation. In that case, you could conflate your measurement and it could lead to a false positive result because changes in your systemic physiology masked changes in your cortical activation. So the idea with short channels is to measure and regress out this component, and it's done by placing a detector very close to the source. In fact, it tends to be an 8 millimeter separation. That's the ideal separation in adults. The separation in infants is 2 millimeters, which is near impossible to do because you'd have to place your source and detector very close together. But short channel measurements are commonly done in adults, and again, you're getting that real-time measurement of the systemic blood flow. Now, you might not have short channels purchased with your system. They do in the NIRx platform. It is a dedicated piece of hardware, but there are analysis methods to address the superficial component. So fear not if you don't have them, but it's certainly a good idea to have them, particularly in cases where your systemic physiology is changing a lot. So say a hyperscanning study with a lot of social interaction, or a motion study where someone's blood flow is increasing a lot. Okay, considerations for fNIRS studies. Signal quality and light penetration. So we talked about signal to noise ratio, SNR, a little bit before, but to hammer down that point a little bit further. The thickness of scalp, skull, and cerebral spinal fluid is not constant throughout the head, and it's not constant throughout development. So over here on the right, we've got images of fMRI scans, and it's reflecting the thickness of scalp, skull, and cerebral spinal fluid on this color scale. So we've got 0 millimeters in blue in thickness down here and 25 millimeters in thickness reflected in red up here. These are 2 adult scans and these are 2 infant scans down here. So with the adult scan, you can see that the central locations on the head have the thickest scalp, skull, and cerebrospinal fluid, whereas the temporal and forehead regions are not nearly as thick. So you're going to get better signal quality in those areas on the whole because more photons are going to be able to pass through to your detector than in areas with higher skull thicknesses, which is going to scatter your photons more. Over here we've got this reflection of these infant measures, and you can see that the scale is much lower, and even still we have much thinner scalp, skull, and cerebrospinal fluid in infants on the top of the head. So they have, you tend to get much better signal quality in infants, and the same also happens in geriatric populations because our scalp, skull, and cerebrospinal fluid thickness decreases. So you tend to also get very good signal quality in geriatric populations as well. It's important to note that density and color of hair can impact optical contact. It's harder for light to pass through darker and thicker hair. So the more sensitive your hardware, the less of a concern this all becomes. So if you have high sensitivity detectors and sources with high source brightness, then you don't have to be as concerned with these physiological challenges. Headgear considerations. First, we'll talk about environmental light. So environmental light is light from any source other than your probes. It could include motion and eye trackers, sunlight, and overhead lights. And if it's not dealt with properly, it can contaminate your measurement. One of the major ways in the setup we can avoid environmental light interference is using a simple, opaque overcap or a shower cap. Overcaps are provided by NIRx. They have zippers on the sides, but if you don't have them, you can also just purchase a $3 shower cap from your local beauty store that has, as long as it's opaque, it'll do the trick. There are other aspects of our hardware that work to keep out environmental light, and we'll talk a little bit about that later on. Another note on headgear, you need to have headgear that's the right size for the participant. If it's poor fitting, it will reduce signal quality. And it does that because it compromises probe orthogonality. That simply means that the probes are not kept perpendicular to the scalp throughout the measurement if the headgear is not the right size for the participant. So headgears must be available in many sizes and fit comfortably for the participant, both for signal quality and for the comfort of the participant. And over on the right, we've got an image of the NIRx NIRS cap sizing chart. So we do offer a variety of sizes for the head circumferences throughout development, having smaller sizes for the preterm and newborn ages, and then larger head sizes for adults. Okay. Probes and optical contact. So with probes, you want to have easy penetration through the hair to make scalp contact, and that's achieved by our probes because they basically come to a point. So it makes for easier parting through the hair. You also want them to be comfortable for subjects. You want the optodes to remain in place for the entire experiment, and you want enough probes to cover the region of interest. So our probes have been really well engineered in order to meet these needs. The last one being dependent on your system configuration. But the other ones, like all our probes, are designed to easily part through the hair and to stay in place for the entire experiment. That's both coming from the probes themselves as well as the NIRS cap accessories that we provide. Okay, movement-related noise. So with fNIRS experiments, you can move, but under the stipulation that the probes remain perpendicular to the scalp and that they move the same amount that the subject moves. When probes shift off the head, it creates a spike or a baseline shift as basically a reflection of the motion artifact. So a spike occurs when the probe momentarily shifts off the head, and a baseline shift occurs when the probe moves off the head and stays there. So these two different manifestations of motion artifact will be important to identify them later on, as well as to address them in analysis. There are ways to address them in analysis, but they're best taken care of in data collection and ensuring that you have your setup to avoid them in the first place. Probes can be made to be more robust movement. Probes, headgear, it can all be made to be very robust to these motion artifacts. And so with generally cable management is one of the most key considerations for preventing them. with proper cable management, these artifacts are generally preventable. So it's very important to review your data in real time as well as later on to get a sense of whether or not your setup is robust to motion artifact. fNIRS limitations. So even with the most sensitive of hardware, you will at some point not be able to image certain layers of tissue in the brain, certainly in adults. So for example, it's not been proven possible to image subcortical structures like the hippocampus with fNIRS. It's just too deep into the tissue and not enough photons can reach that tissue and return back to the detector. fNIRS is known to be most sensitive to the first 15 millimeters beneath the scalp. This can be extended further if you have more sophisticated hardware, but still this is generally the area most people are imaging in with fNIRS. Placement of headgear is a limitation of the methodology, so you need to have consistent placement of headgear across participants in order to ensure that you're imaging the same region across participants. So with our system, our NIRS cap uses EEG 1020 landmarks in order to ensure that you've got consistent placement across participants, and you also have the caps coming in different sizes. But head shape can vary along with head size, so then that will affect also where exactly your probes are imaging. So it's important to keep track of certain fiducial marks, circumferences of your participants. There are simpler methods like simply taking measurements of the participant's circumference of their head as well as measurements from one fiducial to another. There's more sophisticated ways like digitizing the subject. There's a number of ways to do that where you record the location of the optodes and fiducials in 3D space. And you can also go as far as taking an MRI scan and comparing that against your fNIRS activation. So there's a lot of ways to take into account the different head sizes and head shapes. What you decide to do for your lab depends on the scope of your work. But it's definitely something that's important to keep in mind. Result reproducibility and generalization. Reproducibility, how can I reproduce the same results in the same subject? Or can I do so? And the answer is yes. This has been proven by a number of studies. But what's important to keep in mind is that you need to control for anything that can alter blood flow in your participants, like caffeine, exercise, and circadian rhythm. You want your subjects to have gotten a good night's sleep before. You don't want them to have drank caffeine in the last 24 hours or exercise in the last 24 hours. And you actually also want to make sure that you image them around the same time of the day to account for changes in circadian rhythm. Generalization, can my results be applied to the whole population? The important thing to keep in mind here is that hemodynamic responses can vary across subjects and across the head. So that time course that we saw earlier, you're not going to get that exact time course in each subject. Everyone's response is going to be somewhat different, not only in individuals, but within an individual, across their head. So there's more of a reliance on group results for time series analysis approaches like block averaging and SPM and the GLM. Whereas machine learning approaches are going to be better suited for analyzing individual variances. So whether or not your results can be applied to the whole population, it's important to keep some limitations of the fNIRS signal in mind, as well as how you're going to analyze your results. Okay, now to discuss advantages and applications of fNIRS. Subject comfort is a big one. So fNIRS is safe and non-invasive. It has the same form factor of EEG. There's no scalp abrasion necessary, no gooey gel, and you can have very quick setup with it and lightweight headgear. So overall, it's much more comfortable and easier to set up than some other neuroimaging modalities. Portability and hyperscanning. So the fNIRS devices can be portable and wearable, as our NIRS Port 2 device is. You can study real-world environments and tasks with using fNIRS. You can have studies that involve lots of motion, and you can have social interaction studies, so where you're imaging two participants at the same time. So over here we've got two subjects wearing the NIRS Port 2 imaging system, so this would be a hyperscanning study in a real-world environment with lots of mobility available. And over here we've got our lab-based NIRS Scout system. So we've also got a hyperscanning experiment going on here. There's limited motion, but the participants can still move around. Okay. fNIRS applications, so any kind of cognitive research area can be studied using fNIRS. So some examples here: BCI and neurofeedback, cognitive disorders, infant monitoring, social interaction, speech and language, and so on. So fNIRS has been applied in all these various areas. fNIRS lends itself very well to multimodal imaging. That means imaging with multiple modalities. So fNIRS has been combined with EEG, fMRI, TMS, eye tracking, accelerometers, cochlear implants, so on. The nice thing about fNIRS is that the optical signal doesn't interfere with with signals like fMRI and EEG. However, it's not to say that just any fNIRS system is going to combine nicely with any of these systems. Each modality is going to have its own considerations. And furthermore, some modalities from some vendors will combine better with our systems than others. So if you're interested in combining a certain modality with our systems, it's recommended to reach out to our support to get their take on recommendations of systems that partner well with ours. Okay, moving into NIRx product and company highlights. So the difference between LEDs and fiber optic probes. So laser sources and fiber optic detectors, these are passive probes. They transmit photons via glass tubes. The conversion from electricity to light occurs within the main system unit, or the amplifier as it's also known. So over here we've got our NIRScout system. The system itself, that's where the conversion from electricity to light is happening. And the probes coming off here, these are thick fiber optic probes, and they're passive. One thing to note is that for every meter of cable, it reduces the signal by 5%, so there is an attenuation consideration with the length of the probes. And laser sources and fiber optic detectors, they can be fMRI and MEG compatible. So as I said before, not all systems are going to combine well with all other modalities. When you do want to combine with fMRI or MEG, you need to use these fiber optic probes because they don't have any electricity running through them. Okay. Talking about here LED sources and detectors. So in contrast to fiber optic detectors, these are active probes. They're considered active because they have electricity running through them. The conversion from electricity to light occurs at the probe level instead of at the amplifier. LEDs and their cables are much less expensive than fiber optic cables. They're sturdier. They can withstand more kind of intense use because they don't have the glass tubes running through them. They just have electricity running through them. And they can obtain similar and sometimes even better signal quality than the fiber optics. So there's a lot of advantages to having this cheaper, lightweight technology. Okay, talking here about the NIRx short channel bundle. So it's used to measure surface artifact. It divides a single detector into 8 short channel positions. So what happens is you take one of your detectors, you sacrifice it, and you end up getting 8 short channels from it. So here we've got our one detector coming in. It's coupled over here with this canister, and then we have our short channel bundle splitting off into 8 separate detectors. Here we have a close-up view of what the detector looks like underneath the cap. You've got your source right here, and your short channel bundle sits 8 millimeters away from it to then form that short channel. Yes, fits around source with an 8 millimeter separation. Okay, talking about the NIRScout family in a little more detail. So this is our lab-based system. It allows for high density and very versatile setups. The detectors are always fiber optics, but the sources can be LEDs or laser. And high sensitivity avalanche photodiodes, also known as APD detectors, are available. Here we've got a couple different models of the NIRScout. There's the NIRScout, there's the NIRScout Extended, and the NIRScout Extended Plus. So these are basically different sizes of the NIRScout to accommodate different configurations, and it's very easily to upgrade them as your lab, has more demands for fNIRS imaging. Okay, the NIRSport 2 and Aurora. So the NIRSport 2 is our mobile imaging system. It allows for wireless operation or via USB or direct-to-device recording. So there's a lot of flexibility in terms of connection with the data acquisition computer, if any at all. It's the highest quality data from a portable system on the market. We feel very comfortable saying that. It accomplishes this by having high-powered sources and high sensitivity detectors. Accelerometer is available at the probe tip level. So the accelerometer, it has the form factor of a probe, and you can place it anywhere on the head. And similar to the NIRx Scout, it is modular. You can, while each NIRS Sport 2 itself can only accommodate 16 sources and 16 detectors, you can purchase multiple NIRS Sport 2s to increase that configuration up to 64 sources and 64 detectors, as we saw with that video earlier with the 48 sources and 48 detectors. Part of the NIRx platform is the NIRS cap. So part of the NIRS cap is this blank cap. And in addition to a number of accessories and the probes, you end up with your final product, which is the NIRS cap. It's headgear that's going to be very robust to motion artifact when set up properly. It's going to keep probes in place for the entire experiment, and it's going to ensure subject comfort as well as quick setup time, all when done properly. There are many accessories in the NIRS cap. You've got spring tops, which are plastic pieces that have a metal spring in it that push down on the optodes. They ensure that the optodes are able to easily part through hair, and they also come in a variety of tensions to ensure subject comfort. Grommets, standard or spring. So these are going to be either plastic or rubber pieces that are circular. They are embedded in the cap, and they then hold the probe in place in that position in the cap. Labels, red and blue, to easily denote sources and detectors and their numbering within the montage. Optode stabilizers, which are optional. So these are these plastic pieces that sit in between a source and a detector. They keep the optode spacing closer to 3 centimeters, and they also add optode stability to keep them in place for the entire experiment. And finally, your optodes. So with all of these together, we have our headgear that will be used to image our participants. Okay, data validity, signal isolation, and signal system performance in the NIRx platform. Data validity, how do I know that my signal is physiologically valid? Well, NIRx systems have been validated against fMRI in multiple studies. That is the closest we can get to assurance, basically, that the signal we're picking up is physiologically meaningful. Not all fNIRS systems have been validated in fMRI, but NIRx systems have been validated. Signal isolation, how do I know that other sources aren't contributing to my reading? Well, NIRx systems give a dark noise reading at the start of each run. The dark noise is not only a measure of detector performance, but also a measure of environmental light interference. It's basically a reading at the detector when the sources are turned off. So you can tell if you have high environmental light interference. And you can address that by addressing that light source, by using shower caps, and so on. In addition, the NIRx systems are designed to be more robust to dark noise interference. They use continuous wave, time multiplexing, and frequency modulation to further isolate fNIRS sources from environmental light. All of these components in their own way help make the system more robust to environmental light interference. System performance, how do I know that my system is producing clean signals? So here, you want to be sure that your system isn't introducing any variation into the measurements itself. Well, every system is going to introduce some variation, but you want a system that's designed well to minimize that and to be reliable over time. So NIRx systems are extensively tested before they are shipped out to end users, and each system comes with a system performance report, so you know with confidence that your system performs up to specs when it leaves our manufacturing. In addition, users should run the static phantom procedure to test system performance every 1 to 2 months. So every user receives a static phantom brick in which the optodes are placed and then covered with a black laptop case. And then you run a system optimization and you determine the performance of those optodes. So you can say with confidence if your optodes are performing well. We provide a manual with that so you understand the procedure and your results of the procedure. If your optodes aren't performing well, then you can discuss with our support consultants and determine if you need new components. But overall, NIRx systems are quite durable when treated well. They can get many years of use when treated well, both the main system unit and the probes. But you have the static phantom there just to verify that every 1 to 2 months, as well as if you're consistently seeing poor signal quality in some probes, then you're going to also want to run a static phantom test. Moving on to some of our software solutions. The NIRS Lab analysis tool is great for learning fNIRS analysis, piloting, and visuals. So it has a very easy to use GUI that steps through data analysis from just looking at your raw data, looking for motion artifacts, and all the way through statistical analysis using SPM and the general near model. So it is limited in features. It doesn't have robust pre-processing methods. It doesn't even allow short channel regression. However, it does serve a purpose of being a really great teaching tool and helping researchers quickly assess their data. To give some perspective, I myself spent a little over a year in an fNIRS lab, and I struggled to understand analysis in the popular fNIRS analysis platforms like Homework 2 and NIRS Toolbox. But I spent an afternoon with NIRS Lab and it all kind of clicked. So it's a really great resource for students and quickly assessing your data. Here's an example of a visual it creates. So this is the block averaging results of a finger tapping task, showing you activation in each channel in terms of a color map over the course of this block average time course. Okay. Moving on to our data acquisition softwares. So each device has a specific data acquisition software. If you're using the NIRsport 2, you'll image with Aurora, and if you're using the NIR Scout, you'll image with NIRstar. So you'll use one or the other. You won't use both. In addition, also, if you happen to be using the NIRsport 1, which is the predecessor to the NIRsport 2, that uses NIRstar. Neither one is necessarily better than the other. They both produce a number of visuals, and they both offer a number of features like LSL compatibility, triggering via software triggers or hardware triggers. Aurora is just our more recently created software, so the interface is a little more up to date. Okay, the NIRSite montage generation software. So we saw this earlier in the 48 by 48 high density video when the montage was being created. NIRSite allows you to create a custom montage. Now we do offer some predefined montages on our support site, and you're welcome to use those. However, if you want to have a specific arrangement of sources and detectors for your experiment, NIRSite lets you do that. You can use EEG 10.5 positions or arbitrary positions. So if you're not familiar with EEG 10.5 positions, I'd recommend looking into them. Basically, it's a standard coordinate system that can be used across participants based on certain anatomical landmarks of the participant. And you end up creating this grid of dots across the head that correspond to certain scalp positions across participants. With NIRSite, you can import 3D digitized probe and fiducial information, and this allows you to register the anatomical information and probe information of a specific participant to a common atlas. And this will enhance, the number of analysis procedures you can do later on, given that you have information specific to that participant. The ICBM 152 atlas is offered. This is an adult atlas, as well as starting in July 2020, infant atlases are offered. So you have 3D atlas information specific to your population of interest. In the future, NIRSite will include other atlases like the Colin 27 and children atlases. NIRSite saves your montage information in 3D space with this atlas information or your atlas registered information. This saves with your data, and then you can import them into this information into analysis platforms like NIRS Toolbox and Atlas Viewer, which allows you to do things like generate sensitivity profiles specific for that anatomy, as well as region of interest analysis and image reconstruction. So NIRSite, it's pretty easy to use, but it has a number of sophisticated features. And finally, technical support in offices. We like to say that technical support is our most valuable resource. We're here to enable your research. We want to work with you. If you ever have any questions or concerns, please don't hesitate to send us an email at support@nirx.net. We try to respond within 2 business days. We have a dedicated support page with resources such as user guides, documentations, videos, and forums. It's not just information on how to use the device, but information specific to fNIRS research, like experimental design and data analysis and anatomical registration and so on. You can register today, and we recommend you do register today at support.nirx.de. You'll just need the serial number of the device that your group is using. We do this to ensure that only our end users are making use of these resources. We will try and approve you within a business day or 2. NIRx regularly hosts webinars and workshops. We announce these through our social media platforms, Twitter, Facebook. We also announce them through the support site. So if you're registered for this from the support site, you'll stay in the loop on those. In terms of our company presence, we have 3 company offices. 2 of them are in the US, in Minneapolis and New York. And one of them is in Berlin, and the Berlin office is where most of the development occurs. We also have 19 distribution offices worldwide, and that's growing all the time. And we have many end users worldwide in some of the top research institutions in the world. So if you're just joining our family, welcome. You are joining not only a group of dedicated scientific consultants at NIRx who are here to help you, but also a number of neuroscientist researchers around the world. Okay, and this concludes our introductory presentation to fNIRS and NIRx. We hope that you now have a good understanding of the working principles behind fNIRS, some of the challenges and considerations and many applications that fNIRS can take you. Please do not hesitate to send us any questions you have at support@nirx.net, and please ensure that you're registered for the support site at support.nirx.de.